Hello everyone. Welcome to Life Sci Mentors. My name is Dr. Ritu Narwal and today we will study nerve action potential, how the neurons communicate with each other. When a stimulus or signal is perceived by dendrite of one neuron, that information has to be passed through the cell body, then axon and axon terminals so that it can be passed on to the next neuronal cell in order. This communication between two neurons is achieved by generation of an action potential. So now let's study what is an action potential. For any cell to generate an action potential, two things are required. First one is presence of resting membrane potential, and second one is presence of ion channels. So here are two things. First one is what is membrane potential? Membrane potential is the voltage difference across the membrane, which means if you have studied the structure of plasma membrane, one surface of the plasma membrane faces the cytosolic region and one surface faces the extracellular region. So the voltage differs between the inside of the plasma membrane and outside of the plasma membrane, and this voltage difference across the membrane forms the membrane potential. In case of living cells, this membrane potential is generated by flow of ions. Now, another important factor is the presence of ion channels. Because plasma membrane is an insulator, which means unless there is a channel present in the plasma membrane, the ions cannot move across it. Therefore, some gate or some pore or some kind of structure is required for the ions to move past the plasma membrane. Here comes the important factors. Those are the ion channel, which helps in the flow of ions across the plasma membrane. Now let's study the ion channels. There are four main type of ion channels that are found in the membrane. First are the leak channels, then ligand gated channels, voltage gated channels, and mechanically gated channels. Now, what is the function of ion channels? Ion channels helps the ions move across the plasma membrane down their electrochemical gradient. Now, if you see the term electrochemical, you can see there are two parts, electro and chemical. Chemical gradient is across the concentration difference, which means the flow of ions from high concentration to low concentration. And electro part means the flow of charged particles, for example, cations, which moves to the more negative side of the area, and the anions, which moves towards the positively charged area. As ions through these ion channels move through their electrochemical gradient, which means either inside of the cell or outside of the cell, they change the existing membrane potential of the plasma membrane. Now let's first discuss the leak channels. Leak channels are present on the plasma membrane, for example, for sodium or potassium. They can randomly open or close, random open or close. There are more number of potassium leak channels present on the plasma membrane than the sodium channels. And these channels, the potassium ones, are more leakier than the sodium ones, and which makes the membrane more permeable to the potassium ions than the sodium ions. Next are the ligand gated channels, which means they need a ligand for it to open or close. For example, ligands could be neurotransmitters or some hormones. When these ligands bind to their receptor, they change. For example, this is the receptor, and when a ligand bind to it, ये ligand bind करके उस receptor का structure change कर देता है, जिसकी वजह से ये receptor जो है ये open phase में आ जाता है, open stage में, which means now the ions can move past this gate. Then comes the voltage gated channel, which means these gates will open in response to a voltage change in the membrane. These kind of voltage gated channel help in the conduction of action potential in the neuron. Then comes the mechanically gated channels. These channels open or close in response to a mechanical stimulation, which could be in the form of a vibration, touch, pressure. Now let's discuss what exactly is this resting potential. Now let's discuss the resting membrane potential, or how is it generated and what is the resting membrane potential. You already know that this is the plasma membrane structure. This could be the cytosol part and this will be the extracellular part. So when there is a buildup of, for example, positive charged ions or negative charged ion on either side of the membrane, there will be a difference in the charge. For example, in the cytosol part, there are more negatively charged ions, or in the extracellular part, there are more positively charged ion, which means there is a difference across the plasma membrane, which leads to the formation of a membrane potential. Now, this difference in the charge is very close to the surface of the plasma membrane only. Otherwise, inside the cell, the charge is equally distributed. Uh, Again, the more difference between the charge across the membrane, the larger will be the membrane potential. Now, in resting phase, the inside of the membrane is mostly negatively charged, and the outside of the membrane is positively charged because of the presence of more positive charged ions on the outside and the presence of more negative charged ion towards the cytosol side. This condition, where there is a difference in charge across the plasma membrane, this type of cell is called as being polarized. The difference in the electrical charge across the membrane is in the form of potential energy, and it is measured in millivolts. At resting phase, in a cell, there is a membrane potential of minus 70 millivolts, which means the inside of the cell is having more negative charge in comparison with the outside of the cell. So let's now discuss what causes this difference of charge across the plasma membrane. The first one is unequal distribution of ions. Now, towards the cytosol side, we see more number of potassium ions, and towards the extracellular side, we see more number of sodium ions and chloride ions. This is the cationic part. This is an anion. Again, this is a cation. And also phosphates, which are the anions. As we have already discussed the presence of leak channels, which are more in case of potassium, and also they are more leakier than sodium. So what happens is the potassium moves out of the cytosol towards the extracellular space, and the extracellular space then becomes more positive. The second reason for development of this resting membrane potential is inability of these anions to move out of the cytosol. Therefore, the inside becomes more negative and the outside becomes more positive. Then comes the third part, which is electromagnetic nature of the sodium potassium ATPases. Now, these are sodium potassium pumps which pumps out 3 sodium out of the cytosol and brings in 2 potassium inside the cytosol so that the membrane potential is stabilized. But again, the number for sodium is more. Therefore, the resting membrane potential is maintained to be more negative towards the inside and more positive towards the outside. Now let's study the generation of action potential. In action potential, the resting membrane potential changes rapidly and is brought back to normal. This is a sequential event and it is termed as the action potential. Now, there are two phases in action potential. One is the depolarizing phase, second is the repolarizing phase. As I have already told you, a polarized cell is a cell having a membrane potential, which means there is a difference between the number of positive and negative ions inside or outside of the cell. Now, what does the depolarization? The resting membrane potential in most of the cells is minus 70 millivolts. When it becomes more positive or it moves to becoming zero, then positive. This is termed as the depolarizing phase. When the positive voltage of the membrane comes back to zero and then again the negative, this is termed as the repolarizing phase. Now, how are these two phases occurring in case of neuron? Let's understand it. Let's first understand the depolarizing phase. The resting membrane potential is minus 70 millivolts, which means the inside of the cell is more negative as compared to the outside of the cell. Now, when membrane of the axon depolarizes to a threshold which is minus 55 millivolts in response to a stimulus, the sodium voltage gated channels open, which means now there will be movement of sodiums towards the cytosol. Because of this movement of sodium from outside to inside, the inside of the cell now becomes more positive and reaches a membrane potential of plus 30 millivolts. Now, at this stage, let's first understand the orientation of the sodium channel. There are two gates in the sodium channel. One is the activation gate and other is the inactivation. Now let's understand it using the structure. This is the structure of the voltage gated sodium channel. Then this is the inactivation gate and this is the activation gate. This is the inactivation gate and this is the activation gate. At resting stage, when the membrane potential is minus 70 millivolts, the inactivation gate of the sodium channel is open and the activation gate of the channel is closed. Therefore, there is no flow of ions. The ions cannot flow through it. When the voltage changes to minus 55, the activation gate opens and the inactivation gate was already open. There is an inflow of sodium ions towards the cytosol. And when the membrane potential reaches plus 30 millivolts, the gate is closed. This stage is the active stage of the channel. This was the resting stage. This is the active stage of the channel where the sodium ions are moving inside the cell. Now, once the membrane potential has reached plus 30 millivolts, then the gate is inactivated. At this stage, the sodium potassium pumps then moves the sodium from inside of the cell to outside. The voltage gated sodium channels are now inactivated, which means the inactivation gate will close and the activation gate will remain like this only. At this particular stage, the repolarization phase begins. To summarize the depolarization phase, the membrane potential was minus 70. This, which in response to a stimulus was towards the positive side and was minus 55 millivolts. Then voltage gated sodium channel opens, because of which the inside of the cell becomes more positive and reaches a plus 30 millivolts. Now, the voltage gated sodium channel has three orientations two gates. One is activation gate, inactivation gate. Now, when the voltage gated sodium channel is at a resting stage, which means when the membrane potential is minus 70, the activation gate and the inactivation gate of the voltage channel is closed, which means there is no inflow of sodium. When the threshold, which is minus 55 millivolts, is achieved by the membrane, then the activation gate of the voltage channel opens, which then through which the sodium ion then enter the cell from outside to the inside, making the cell more positive, which means at this stage the membrane is depolarized, which means the membrane is more positive towards the inside as compared to the outside. At this stage, when the membrane reaches plus 30 millivolts, the gate is inactivated, which means now the inactivation gate will be closed. At this particular time, the sodium potassium potassium pumps also works, and at this point, the sodium potassium pumps also works and moves the sodium ions from inside of the cell to the outside of the cell, which begins the repolarization phase. Now, the membrane has reached a potential of plus 30 millivolts, and it has to go back to its resting membrane potential, which is minus 70 millivolts. Now, how it will achieve it? It will achieve it with the help of potassium voltage gated channels. At this stage, the potassium ions will now move out of the cell. This is E, this is cytosol. The potassium ions will move out. Now, because the sodium gate has inactivated, the sodium inflow will be closed. There will be no movement of sodium ions inside the cell. There, therefore, now the membrane becomes more negative towards the inside as compared to the outside, because the potassium ions move out of the cell and there is no inflow of sodium ions. Therefore, more positive charge is moving outside, which brings the membrane potential to minus 70 millivolts, and this is the repolarizing phase. Now let's understand this action potential by a graph. On the y-axis, we have the membrane potential. On the x-axis, we have the time. Now, if I mark, if this is the resting membrane potential, this is the resting membrane potential, minus 55 millivolts is the threshold, which means the action potential will initiate. Then it reaches a plus 30 millivolt stage, which is something like this. Now, when it reaches plus 30, this is the depolarizing phase. From minus 70, the membrane potential has now become plus 30 millivolts. Now, the membrane potential, once it reaches plus 30, it has to then again come back to minus 70. This is the repolarization phase. Sometimes the threshold, sometimes during the repolarization phase, the membrane potential goes up to minus 90 millivolts. That is also termed as the hyper repolarization phase, but then it again comes back to the resting membrane potential, which is minus 70 millivolts. Now, again, to summarize the action potential, when this is the resting membrane potential, which is minus 70 millivolts, when the axon membrane receives a stimulus up to which brings its voltage to a threshold of minus 55 millivolts, the membrane starts depolarizing, which means opening of the, which means inflow of the sodium ions. Inflow of the sodium ions from outside to the inside of the cell, making inside more positive, making the voltage more positive. It is moving towards the positive side. All right, this is the depolarizing phase. Now, when it reaches plus 30 millivolts, then begins the repolarizing phase. In the repolarizing phase, what happens is the sodium inflow stops and the potassium outflow begins, which means now the inside of the cell will become more negative and the outside will become more positive. Then the membrane potential now comes back to its resting phase, which is it will come back to its resting phase, which is minus 70 millivolts. Now, at this stage, if I mark this area with yellow, this area, this is called as the absolute refractory period, which means when the membrane is in the depolarizing and the repolarizing phase, there will be no other action potential that can be generated regardless of the intensity of the stimulus. Which means कितना ही stimulus आ जाए, strong stimulus आ जाए, but second action potential will not be generated when the neuron is already in its depolarizing and repolarizing phase. Other than the absolute refractory period, there's another phase also. This period is called as the ref, relative refractory period. During this period, another action potential can be generated, but with a stronger stimulus. So this was all about the action potential, how the neurons communicate with each other. Thank you so much.